faithfully delivering value and exploring great books worldwide. Hi, this is Sally with Book Talks. Today we're interpreting gene society. Humans form societies, animals have theirs, so can genes form one too? So, what rules do genes follow in this book? It reveals the cooperation and competition within the gene society. The book is co authored by Professor Itai Yanai from the US and Professor Martin Lercher from Germany. Professor Yanai is a scholar at Harvard's Radcliffe Institute, teaches at NYU School of Medicine, and heads their Computational Medicine Institute. Meanwhile, Professor Lercher teaches bioinformatics at Heinrich Heine University in Düsseldorf, Germany. Both are top experts in bioinformatics. They're experts on genes and keep up with modern advances. Together, they present a compelling theoretical framework in this book. They see genes as tiny members in a vast society, connected, cooperative, yet competitive and adversarial. From there, they unveil the evolution of human genetics. Now, I'll break it down into two main sections to cover the book's essence. First, how genes control our bodies. Second, how the gene society functions. Starting with the first question, how genes control us. Genes are vital. They're inside us, holding genetic info that defines traits like height, skin tone, hair length, eye size, and more. So, what are genes really? To be specific, our bodies have cells, each with a nucleus containing DNA molecules, long ribbon like strands. Genes are how these DNA molecules are arranged and combined. Think of DNA as a super long piece of paper. On this paper, there are special codes that record details about biological traits. This info is spread out across the paper. For instance, hair color might be noted in one spot and another trait far away. This record might describe the number of fingers. Each such record corresponds to a gene, and all human genes are concentrated on 46 such strips, meaning 46 DNA molecules. Every cell contains a set of these DNA molecules that control its life activities. The cell is the domain governed by this DNA set, and its direct jurisdiction ends there. Theoretically, the DNA molecules of any individual are identical, carrying the same genetic information. That's because humans develop from a fertilized egg, and all other cells' DNA molecules are produced through division from the single egg cell. Like ancient manuscripts, all cellular DNA is transcribed from the fertilized egg's DNA. How enormous is this transcription task for the fertilized egg's DNA? Each human cell contains over 20,000 genes, and a human body has about 100 trillion cells. Copying all these genes precisely is staggering. This can be described as a colossal undertaking. Facing such massive data replication, even the most advanced computers can't guarantee 100% accuracy. So errors inevitably occur during DNA transcription. If a segment becomes flawed and unreadable, the gene fails. Or if the segment's meaning changes, the gene's function alters. These changes directly affect both the cell containing the gene and all its descendant cells. So, in other words, more transcriptions or cell divisions mean more accumulated errors and changes. Cancer develops through this very process. Cancer cells escape bodily control, divide endlessly, eventually invading space, hijacking nutrients, causing death. To become this rampant, a cell must break 8 locks, 8 critical mutations, safeguards set by natural evolution. But locks eventually open. As the book says, live long enough and cancer becomes inevitable. Not all mutations cause trouble. They let us see a vibrant world, while the world seen by most mammals is way more monotonous than what you see. That's because humans gained stronger color vision through gene mutations. The gene responsible is called the opsin gene, and this is pretty much certain. In ancient times, animals' DNA had just one such gene, allowing only light-dark changes, basically black and white vision. But through long evolution, during DNA transcription, the opsin gene segment got duplicated, boosting its number to 5 at one point. Then, in later transcriptions, errors made those 5 genes all different. One became the rhodopsin gene, can't see colors but sensitive to dim light, giving animals night vision, meaning they see clearly at night. The other 4 are cone-opsin genes, each detecting a color for rich hues. At this point, the opsin genes formed a gene family. Sadly, during mammal evolution, 2 of the 4 cone-opsin genes got lost in transcription, so most mammals only see 2 colors. Cats are dichromats, seeing things like human red-green colorblindness. Later, primates, the branch humans are in, accidentally duplicated another cone-opsin gene. After this mutation, we gained a third cone-opsin gene, ending up with red, green, blue trichromatic vision. Though our color sense isn't as good as birds and other tetrachromats, it's better than those with only 2 color vision. This is thanks to that cone-opsin gene duplication. As biologist Suzumu Ono said, natural selection just tweaks redundancy, while duplication is true creation. Besides passive mutations from transcription errors, some genes can change actively. In the human body, B cells and a few others can edit their own genomes. B cells are immune cells that make antibodies against pathogens, specifically wiping them out. This requires them to edit their own antibody genes to design specialized antibodies. It's precisely this principle that led humans to develop vaccines. Vaccines are pathogens that have been rendered harmless. After injection, they train the body's immune system. However, changes in B cells only affect themselves and don't alter genetic material in reproductive cells. Meaning, if you get a smallpox vaccine, your child still needs one. This immunity isn't inherited. Now we understand genes. They carry genetic information, determine who we are and our appearance. They transmit info through copying, but errors can occur. That's mutation. Gene mutations might bring disasters. Cancer is the malignant outcome of gene mutations. On the flip side, mutations also bring benefits, like letting us see a vibrant world. We owe this to gene mutations. Throughout human evolution, genes have constantly changed, accumulating over time to gradually transform our bodies. So how do genes function in the body? Genes can't act directly. They need tools to control the body and only activate under specific conditions. In the genetic factory, DNA is like a supervisor issuing instructions. RNA is the messenger, carrying genetic info from the nucleus to guide protein synthesis elsewhere. This biological principle is called the central dogma. Actually, at the start of each gene is a component called a promoter. It's like a switch determining when and where the gene activates. Only when activated and producing corresponding proteins can a gene fulfill its function. Let's consider an example of gene function. Scientists identified a gene called FOX2 in families with language disorders. In affected individuals, this abnormal gene causes inability to understand or use language. Such cases are incredibly rare. All humans have near identical sequences for this gene, meaning it's super important. If it gets miscopied, natural selection usually wipes out those flawed individuals ruthlessly. Unlike chimpanzees and other apes, in humans, the FOX2 gene is highly active in a brain area called region X. This region is thought to handle language. Exactly because this gene is active in region X, it gives us way richer language than other apes. And hey, the same thing is seen in birds. Compared to dull birds, singing ones have their FOX2 gene active in region X, and canaries change their songs in specific seasons. Only then does the FOX2 gene kick in. From this, we infer that changing when or where a gene activates can alter biological functions. No new genes needed. It's just that gene regulation shifts. This is like every team having the same resources. Only by using them smarter do you get good results. So that covers how genes control our bodies. Next up, how the gene society operates. That's the book's core question. Inside us, tens of thousands of genes interact in complex networks, pull one thread, and the whole thing moves. Coming up, How the gene society formed and its four hidden rules. First off, how the gene society came to be. Many literary stories mention a species, a human-ape hybrid called Shingren, using Shing for gorilla, meaning human. Sure, these tales can be deeply moving or totally sensational, but honestly, such creatures just aren't possible. Even though we know humans and chimps shared a common ancestor millions or more years ago, humans and chimpanzees have only a 1% difference in DNA sequences. Yet, humans and chimps can't produce hybrid offspring anymore. Biologists call this reproductive isolation, meaning they can't breed or offspring are infertile. So reproductive isolation stems from a core evolutionary principle. Every genetic change must never harm its carrier. If a mutation harms its carrier, natural selection easily eliminates those with the flawed gene. The result? That gene itself gets wiped out. Therefore, when a gene mutates, survival depends on integrating into the existing genetic community. Over millions of years, countless new genes emerged in human genomes, continually joining and reshaping their gene societies. The same happened with chimpanzees. Today, humans have their gene society, chimps theirs. When they meet, only system conflict errors occur. While many genes match between systems, differing ones alter each society's composition, making them incompatible. Within gene societies, genes cooperate while competing, forming unspoken rules. Let's go through them one by one. First rule: use sexual reproduction to filter superior genes and purge inferior ones. Undoubtedly, the emergence of sexes revolutionized biological evolution. Genes from both parents undergo fully random recombination during reproduction. This process combines quality genes while efficiently clearing harmful mutations from the gene society. Individuals with harmful mutations lose in species competition, failing to reach adulthood or find mates. With low reproduction rates, harmful genes vanish from society when these individuals die. Thus, sexual reproduction became an efficient gene screening model, dramatically accelerating evolution. Thankfully, most bacteria lack sexual reproduction. Otherwise, they develop drug resistance faster than pharma companies make new meds. However, sexual reproduction also creates problems. That's genetic dilution. Children are like joint stock companies of their parents. Each parent only holds a 50% stake in their child. Nearly every paternal gene pairs with a maternal counterpart. They check and balance each other, jointly determining the child's traits. Such balancing pairs are alleles, like whether you have double or single eyelids, decided by one allele pair. Only half of each parent's genetic material passes to offspring. Pass it down to grandchildren, and only a quarter remains, decreasing further. To counter this dilution, the Gene Society devised a compromise. During germ cell production, genomes hold a lottery via meiosis, reshuffling genes, discarding half to create balanced cells. This lets superior genes survive undiluted. In such a complex gene society, a single gene's power is limited. Thus, the second hidden rule: genes form small groups for mutual support. These groups gain evolutionary edges over solo genes, cementing their status. Take one group. Normally, three genes break down a protein, preventing brain cell buildup. If one gene fails, junk proteins flood brain cells, impairing function and causing neural loss. That's how Parkinson's disease happens, as we know it. Meanwhile, genes near each other on DNA can form alliances. During germ cell production, they're likely are assigned together, so strong genes shield nearby ones, even imperfect ones. The SRI gene shields almost the entire Y chromosome. You probably know the Y chromosome is a human sex chromosome. So you might have guessed, this gene determines human sex. Having it makes you male. Except for a region on the Y chromosome with about 20 genes, the rest doesn't participate in genetic reshuffling during germ cell formation. They're permanently locked in this genetic combination. Even with minor mutations in any gene, the whole set barely holds. But when a catastrophic mutation threatens the whole group, it's either total collapse or kicking out the mutated gene. So the smart choice was made. The Y chromosome kept shrinking, from thousands of genes initially to under 200 today, sacrificing minor genes to keep the core team stable. This might be the Gene Society's version of sacrificing pawns to save the king. Beyond forming small groups, gene cooperation shines brightest in cellular evolution. That brings us to the third hidden rule: mutual bundling to integrate premium genetic resources. It's brilliant cooperation, genes of diverse origins, sharing resources to create entirely new cells. Long ago, no animal, plant, or fungal cells existed, just archaea, bacteria, and cyanobacteria, simple single-celled beings. Archaea were large engulfers but poor at oxygen-based nutrient breakdown, but bacteria mastered oxygen use, while cyanobacteria could photosynthesize. So, the tale began when an archaeon swallowed a breathing bacterium. Instead of destroying it, the archaeon housed it like a tenant to process nutrients. They formed a shared destiny community in one evolution's battle. That breathing bacterium became mitochondria, one of today's crucial cell structures. Mitochondria governs cellular energy supply. Even now, mitochondria retains some genetic material, while another part merged into the archaeon's genome, specifically in today's nucleus. Some archaea then swallowed cyanobacteria, gaining photosynthesis. These evolved into plants, the structures performing photosynthesis. Well, chloroplast is the name the cyanobacteria evolved into after billions of years. That's the grand cooperation we explored. Now, let's examine the fourth hidden rule. Junk genes survive through parasitic means. They're special DNA sequences that can loosely be called genes. Though they contribute nothing to the gene society's success, they have survival tricks to make cells copy themselves. Then, using proteins they produce, they slice DNA and splice their copies in, achieving genetic copy-paste. Imagine if only one such gene existed. A mutation could disable it, turning it into pure junk, redundant DNA. But with many copies, they avoid elimination by such accidents. Instead, they can build a family mass-producing active junk genes. If they multiply faster than being cleared out, the family survives. Now, with 500,000 copies in the human genome, they're remarkably successful. Sure, they burden cells, but unless they overwhelm the system, they'll persist. In this section, we covered the 4 hidden rules. Sexual reproduction helps the gene society select the fittest. Gene society cooperation involves forming small groups and integrating quality resources. Junk genes survive through parasitic tactics. This is the gene society, a complex system where genes constantly change, accumulate variations, and drive evolution. They regulate life traits through cooperation and conflict. They also exploit rule loopholes, seize opportunities to grow, clear out weak members, and pass down through generations. All right, that concludes our interpretation of gene society. Check the listening notes below the audio and see you tomorrow.